This is lecture E on the hypothalamal pituitary gonadal axis, and we now are asking questions about the regulation of the gonadotropins, in other words, the gonadotropin genes. In this lecture, we will discuss the promoter regions of these genes and how the expression is regulated. So we recall that the gonadotropin genes are of two types because the gonadotropins, both FSH and LH, are heterodimers with an alpha subunit that's expressed in the gonadotropes and the thyrotropes and the placenta. Remember, the placenta is a source of chorionic gonadotropin, and therefore also has the alpha subunit. The beta subunit is restricted to certain cell types. The FS beta subunit gene is expressed in the gonadotropes and affected by activin, inhibin, and GnRH. The LH beta, on the other hand, even though still expressed in the gonadotropes, has an expression that is dependent upon GnRH. And as we'll see, it is affected by at least androgen negative feedback. The chorionic gonadotropin beta is a separate gene that evolved from LH beta gene and is selectively expressed in the placenta and is present within primates and equids. This would be horses. So the tissue and the cell-specific expression of the LH beta gene will be the point of our discussion in this slide. And throughout, we'll just focus on that primarily. Again, the LH surge is very important in the menstrual cycle, and we somewhat keep a focus on that idea. Not that we're going to discuss how the LH surge is regulated through the gene expression of the LH beta subunit in this context, but a reason for our focus just on that gene. So there is a GnRH specific element that has been detected through analysis of the promoter region of the LH beta gene, and actually there are two of them. There is an interaction with SF1. We recognize that. We'll talk about it a bit more in a moment. And it's responsible for both basal and these two collectively are responsible for basal and GnRH stimulated transcriptional activation. There's an EGR, an early growth response protein 1 binding site nearby as well, or a consensus sequence. And we see again that there are two of these GSEs, a proximal and a distal, and we'll go into a bit more detail on those two as we proceed. The locations of these are shown here, minus 59, minus 50 for the EGR, minus 59 for the proximal GSE, minus 113 for the distal EGR, and minus 127 for the distal GSE. So they're really not that far apart. So they're even though these are referred to as distal, they're not extremely, thousands of kilobase pairs away. The steroidogenic factor 1, the SF1, as you recall, is an orphan of the ligand-dependent nuclear hormone receptor family, and 25-hydroxycholesterol is a possible ligand. It's expressed in the adrenal gland, the gonads. Here, of course, we talked about the role in the expression of the enzymes responsible for steroidogenesis, and we see commonly then for expression of the hormones too that drive the adrenal, at least the gonads anyway, the gonadotropes. The tissue-specific gene expression is mediated through this, as we recall, because the SF1 gene knockout will lack the expression of the LH beta, the FSH beta subunit genes, and the GnRH receptor gene. So this is where we saw this information previously. There's a decreased alpha subunit mRNA as well. So the alpha subunit is not entirely dependent upon the SF1, whereas the others listed here are dependent upon the SF1 binding to the promoter region. In vitro transfections have shown that the AF1 binds to the LH beta gene, and there is a physiological significance to this in that if transgenic mice are produced with the LH beta promoter, including both the proximal and distal GSEs, and use a cat reporter, then there is normal cat expression meets the expected level of expression is what's meant there by normal. The alternative type of promoter would have a GSE mutation on the LH beta promoter, again with the cat reporter, and there is a decreased cat, but it's not eliminated. So in vivo there may be something else that contributes to the expression. And that may indeed be the early growth response protein, the EGR1. It's also known as NGF1A, nerve growth factor 1A, ZIF286, or CROX24. So 4 names for the same protein. It is a member of the immediate early gene family, as is the SP1. There is a zinc finger domain for that type of interaction with the DNA at the consensus sequence CGCC, either C or A, and CGC. It's expressed in gonadotropes and many other cells. The EGR1 knockout shows a loss of the LH beta expression. So again, the LH beta expression is dependent on EGR1, as it was the AF1, and the FSH beta expression is maintained. Let's now think a little bit about the gonadotrope-specific element, the GSE, and we see that it resembles a nuclear receptor binding haft site, probably monomeric binding to the haft site. SF1, EGR1 heterodimers would bind after the EGR binds to the DNA in this region. So since this earlier work, there's been quite a bit gained about understanding these GSEs, and the nomenclature has changed a bit, as you will see. So there's a more recent article on this topic, the small nuclear ring finger, or SNURF, SNRF protein, stimulates the rat luteinizing hormone beta promoter by interacting with SP1 and SF1 and protects against androgen suppression. And this is in the Molecular Endocrinology 2004, volume 18, issue 5, page 1263. So this is an image of the distal of the promoter region for LH beta, showing the distal and proximal GSEs. So here's the start site shown here, around plus 44, and we see that there, near to it, there is the proximal GnRH response region. So a slight change from this GSE over a few years. And minus 112 to minus 60, so it's a little more specifically clarified at this time. Minus 246 is shown here, and then minus 350 to minus 456 is the distal GnRH response region, and we see they contain SP1 binding sites and SF1 binding sites, as well as the ERG1 shown here. So indeed, you're recognizing that the prior description was focusing primarily on this region, and now even a more distal region that is important for both basal and GnRH activation of the LH beta subunit. So I'm sure there's many research articles in between that clarified all of that, that we've jumped across to get to this more recent one. So another thing to note about this complex is the androgens suppress the LH beta gene subunit gene expression through interference with binding to activating transcription factor 1, the oh, excuse me, these aren't shown on here at the moment. The ATF1 and C-Jun to the promoter. So there's additional binding sites, of course, than what are shown here. And the ATF1 and the C-Jun are important for maintaining expression when negative feedback is not occurring at a significant level. When androgen levels are high, for example, in the males, then it'll bind to its receptor and interfere with the binding of ATF1 and C-Jun to the promoter, and thereby repressing the gene or reducing the level of activation, I guess would be the way to think about it. So the androgen receptor does that in this fashion rather than binding directly to any DNA in the promoter region, such as an androgen response element. Therefore, SNRF is a coactivator, and it prevents the androgen receptor from disrupting the recruitment of the transcription complex after androgen treatment. So the SNRF plays an additional role in balancing the degree of negative feedback. This slide is showing the SNRF and the ring finger and the AR androgen receptor interacting domains are shown here, and they are required for the full SNRF coactivation of the LH beta promoter. So we see the N-terminal and the carboxy terminal. It's about 194 amino acid residues. Toward the N-terminal end, there is a DNA binding domain. And if you read this article, while you're reading this article, you'll see that they've either mutated or deleted key amino acid residues or substituted them to determine and to identify the important sites. The ring finger domain shown here, SP1 binding shown in this area here, and the androgen receptor binding region shown here. So when the androgen receptor is ligated, SNRF can bind to the ligated androgen receptor and keep it from interfering with the binding of other activators such as the ATF1 and the C-Jun that we had mentioned. So this illustration then is from the figure 9 in that article. It's a model for the SNRF interaction with the two GnRH responsive regions of the LH beta promoter. The top panel here illustrates the distal and the proximal GnRH response regions of the LH promoter, and of course the transcription factors proposed to bind to these regions. SNRF has been shown to interact with the SP1 and SF1, as we said, and it's proposed that the DNA between these two, the distal and the proximal regions, forms a loop, as we can see in the lower panel, enabling the interaction between these two regions. Excuse me. The SNRF binding with factors in both the regions may enable it to mediate the interaction between the regions, thereby increasing transcription under basal and stimulated conditions, as we had said before, and actually preventing the androgen receptor from interfering with this complex formation that brings in the transcription binding protein complexes for sustaining basal and allowing response in the expression to the GnRH. So here you can see in this case when GnRH and the androgen are added, you get this, it's proposed that you get this type of a complex formation that promotes the expression of the gene. Well, let's exercise our minds a bit with these final few slides. and ask ourselves some questions. How could we differentiate whether GnRH increased cyclic AMP or increased PKC? In other words, using forskolin and a 4-ball ester. Remember, the forskolin will increase adenylate cyclase and the 4-ball ester will activate the PKC. Increases the steady state of the LH-beta messenger RNA by increasing transcription or decreasing turnover. So you would block transcription with actinomycin D to determine whether the increased steady state excuse me, of the LH-beta messenger RNA was transcription dependent or mediated through the decreasing turnover rate of the message once it's in the cytoplasm. So this would determine the rate of decay. Subsequent to blocking transcription, you would just determine the rate of the decay and see if under these stimulated conditions whether you had an extended or increased, a decreased rate of decay. Right? Decreased rate means that it would hang around longer. In other words, an increased steady state of the LH-beta and messenger RNA. So if there is, if you're able to detect higher levels, steady state or stable higher levels of LH-beta mRNA when GnRH is acting on the gonadotropes, you're asking whether it's mediated through the cyclic AMP or PKC second messenger systems. Then what you need to do is to block the transcription and determine the rate of decay. If the rate of decay remains not significantly different from normal than it was depending upon transcription. If the rate of decay is significantly less than the normal rate of decay, that means that the message is sitting in the cytoplasm longer and you're not having a need for the contribution of new message to sustain the higher levels, the steady state, increased steady state of the message in the cytoplasm. Another approach could be transient transfection using, for example, the GGH3 stably transfected cells that express the GnRH receptor, or alpha T3-1 gonadotrope tumor, and test two regions of the promoter necessary for the expression by the GnRH that would be targeted by either of the types of transcription factors known to be affecting the expression related to the prior two second messenger systems. And you can use an electrophoretic mobility shift assay to see if nuclear proteins are binding to either of these under the stimulated conditions. Then a high pulse GnRH frequency or constant low amplitude would produce homospecific desensitization and also downregulates the alpha and the beta subunit expression, and then ask what of the second messenger systems for the LH beta increased cyclic AMP increases the LH beta messenger RNA, and increased PKC also increases the LH beta messenger RNA. So it seems that the GnRH receptor may be driving LH beta expression through these two mechanisms. And we end our discussion